Welcome to this explainer. We're jumping right into it today because we are exploring the fascinating and honestly incredibly complex world of epigenetics. If you've ever wondered how the exact same DNA can create a skin cell in one part of a biological system and a brain cell in another, well, you are in the right place. We're going to look at a revolutionary technology that is completely changing how researchers decode the genome. To get us started, think about this. What actually controls the software of the genome? We all know DNA is the hardware, right? That foundational sequence of A's, C's, T's, and G's. But hardware doesn't do a thing without software telling it what to run. So how are specific genes turned on and off without actually changing the underlying DNA sequence? That's the magic of epigenetics. And to really grasp it, we have to look at its primary mechanism. And that mechanism is DNA methylation. It's essentially the vital molecular switchboard for biological systems. By adding tiny methyl groups to DNA, specifically hitting those cytosine bases, it regulates gene expression, dictates cell differentiation, and drives development. Getting an accurate read on these methylation markers is absolutely crucial if we want to understand the molecular mechanisms of life. But historically, detecting these tiny modifications has been a massive headache. For years, the gold standard for mapping these markers was Whole Genome Bisulfite Sequencing, or WGBS. But I want you to think of WGBS like a sledgehammer. It relies on harsh chemical reactions and extreme temperatures to convert unmethylated cytosine into uracil. Sure, it works, but it literally causes massive DNA damage and degradation in the process. You end up losing a ton of vital information. On the flip side, we have EM-Seq, Enzymatic Methylation Sequencing. This is the scalpel. It uses a gentle, super precise enzymatic process that preserves the structural integrity of the DNA while mapping those exact same methylation sites. What makes this transition so revolutionary is the sheer difference in the starting material required. Because that WGBS sledgehammer destroys so much DNA, it usually requires a massive input, around 1,000 nanograms or 1 microgram of DNA just to get the protocol running. But the EM-Seq scalpel, it only needs a tiny 10 nanograms. To put that into perspective, that's a total game changer. For anyone working with incredibly rare, microscopic, or fragile samples, this dramatically lowers the barrier to entry. Section 1. How EM-Seq works. Molecular Tag Team. Okay, let's dive into exactly how this works. This is where the magic happens. Step 1 relies on an enzyme called TET. TET is basically an oxidation booster. It seeks out the methylated cytosines, those 5-MC markers, and oxidizes them. This effectively throws a chemical shield over them, keeping them safe from what comes next. Then, in step 2, the APOBEC enzyme tags in. APOBEC is a deamination executor. It targets all the regular, unmodified cytosines and converts them into uracil. But remember, those methylated cytosines are shielded, so they stay perfectly intact. Finally, in step 3, standard PCR amplification converts the uracil into thymine. When you sequence it, you just compare it to the original, and boom, you see exactly where the methylation occurred. It's a stunningly elegant biological tag team. Because of this gentle enzymatic approach, EM-Seq brings some absolutely massive technical advantages to the lab. First off, it preserves complete DNA integrity. Since the DNA isn't getting chemically shredded, EM-Seq generates longer, more intact library fragments, which minimizes those frustrating sequencing gaps. It also completely eliminates GC coverage bias. WGBS famously struggles with genome regions rich in guanine and cytosine, which creates blind spots. EM-Seq, it gives you uniform coverage across both GC-rich and AT-rich areas. And because of that superior coverage, you uncover significantly more CpG islands, meaning you aren't missing crucial data. Section 2, Advancing Biological Research, Broad Applications. In foundational biological research, EM-Seq is proving invaluable for studying highly fragmented, trace amounts of DNA. Researchers are constantly looking for non-invasive ways to extract profiles from cell-free DNA circulating in an organism's system. But these fragments, they are incredibly microscopic. Because EM-Seq only requires that tiny 10 nanogram input we talked about, researchers can successfully extract deep methylation profiles from these precious trace cell-free samples, paving the way for vital biological discoveries. And this brilliantly illustrates the impact on developmental biology too. When researchers study the early stages of life, tracing how a single cell decides to become a specific tissue, they're working with microscopic, highly precious samples, like embryos, oocytes, and early stem cells. You simply cannot get a microgram of DNA from these sources to run old-school WGBS. EM-Seq empowers developmental biologists to track dynamic epigenetic changes without needing massive sample volumes.
But hey, it's not just about animal biology. In botany and agricultural science, researchers are using EM-Seq to map complex plant genomes. When crops face environmental stressors, like a sudden drought, extreme cold, or pests, their DNA methylation patterns change dynamically to regulate growth and survival. By analyzing these shifts with this level of precision, agricultural scientists can unpack the epigenetic mechanisms of stress resistance, which ultimately helps engineer much more resilient crops.
Section 3. Overcoming sequencing challenges, the in-house bottlenecks.
Now, this technology is undeniably powerful, but attempting to scale this up and run EM-Seq entirely in-house, that comes with some massive technical hurdles that can seriously bottleneck your research. Consider the common roadblocks. First, DNA extraction and fragmentation. If you don't use the exact right lysis buffers or ultrasonic crushing methods, you literally lose your already microscopic sample before you even start. Then comes library construction. Inefficient linker connections give you PCR bias, totally ruining your data uniformity. But honestly, the biggest bottleneck is the bioinformatics. EM-Seq generates a staggering volume of data. The computational resources required for memory, storage, and parallel computing just to analyze sample heterogeneity, it can completely overwhelm a standard lab's IT setup. This is exactly where researchers need a frictionless partner to shoulder the technical burden. Enter CD Genomics. Their entire mission is to harness the power of EM-Seq technology to deliver exceptional end-to-end methylation analysis services. So instead of fighting with low-yield libraries and server crashes, researchers can leverage a specialized team designed to solve these exact bottlenecks. Section 4. Your EM-Seq service partner, turnkey research solutions. CD Genomics essentially provides a seamless journey from start to finish. You begin with sample submission, following their strict guidelines to ensure maximum integrity. From there, their experts completely take over. They handle the meticulous DNA extraction, that super delicate TET and APOBEC enzymatic library prep, and they run it on high-throughput next-gen sequencing platforms, guaranteeing a coverage depth of at least 30x. And finally, they do the heavy lifting on the data analysis side, handing you a comprehensive bioinformatics report. So the crucial takeaway here is this. You don't just get a hard drive dumped with raw, unreadable FASTQ data. No way! You receive customized, highly structured bioinformatics reports that are ready for immediate analysis and publication. Their team maps the overall methylation levels, performs deep site-specific comparisons of differentially methylated regions, visualizes everything through PCA clustering and heat maps, and even conducts KEGG enrichment pathway analysis. They take raw sequencing reads and translate them into actual, actionable biological insights. Which brings us to a final thought. Are you ready to unlock the epigenetic secrets hiding in your samples? Epigenetics really is the frontier of biological research right now, and EM-Seq is absolutely the ultimate tool for that exploration. By partnering with experts like CD Genomics, you get to bypass the frustrating technical hurdles and focus entirely on the science, uncovering the hidden software that drives the genome. Thanks so much for joining me on this deep dive, and keep exploring.